Hi, everyone. Thank you all so much for joining us for the second webinar in our series hosted by NHDES and follow-up to the release of the New Hampshire Cyanobacteria Plan. My name is Kate Hastings. I am the Cyanobacteria Harmful Algal Bloom Program Coordinator here at NHDES, and today I'm going to be presenting on some cyanobacteria basics, how to do a self-risk assessment, and how you can help with our monitoring program, and I'll end with a review of cyanobacteria trends in New Hampshire over time. A couple of housekeeping items before I get started. This webinar is being recorded, and we will share a link of the recording by email to you, and it will also be posted on the NHDES website in a couple of days. All attendees are in listen-only mode, which means you don't have access to your microphones or your webcams, but you can submit questions that you may have during the presentation through the question box that you should see on your screen. So I plan on presenting for around 40-45 minutes today. At the end of that, my coworker Michelle Condon will join me to help moderate the question and answer period for the following 15 to 20 minutes. If we don't get to your question during that time, I will email you with a response. So just sit tight for that if we don't get to your question. And with that, we will get going here. If I can figure out how to do this again. Here we go. Okay, so starting with the basics, cyanobacteria are a type of very diverse group of organisms that fall under the larger category of phytoplankton. And phytoplankton are even more diverse, larger group of organisms, including dinoflagellates, diatoms, chrysophytes, and green algae. And all of these are the primary producers in our aquatic ecosystems. So they're very important, the basis of the aquatic food webs. And typically they all exist in balance with each other. They take turns through the growing season, being more or less dominant than the other organisms. They all require the same components to grow and photosynthesize, so access to nitrogen and phosphorus nutrients, as well as access to sunlight. However, if there's an imbalance in either of these inputs through excessive nutrient loading or through climate change, it tends to be cyanobacteria that takes advantage of that imbalance and grows out of control into what we call blooms. So why do we see cyanobacteria bloom more frequently than other phytoplankton? So to give you some perspective, they've been on this planet for around 3.5 billion years, so they've had a long time to perfect their strategies, and we can thank them for oxygenating our planet. So they have a handful of what we call competitive advantages that give them the edge over other phytoplankton. So one of these is a set of accessory pigments on their chlorophyll called phycobilins that allow them to harness a greater range of sunlight to perform photosynthesis, so they can perform better in low light conditions than other phytoplankton. It's also these pigments that give them their name. So cyan is for the blue-green color that you see in this picture, which is when the chlorophyll degrades, the cyan pigment is left, the phycobilins are left. So cyanobacteria is just the color cyan, blue-green, O bacteria, just to demystify that name a little bit. So another advantage that cyanobacteria have is many of them can control their buoyancy. So they can move down in the water column to access nutrients or up in the water column to access sunlight based on their current needs. Many of them also have specialized cells. Some of these are called heterocysts that allow them to fix atmospheric nitrogen, tapping into a resource that other phytoplankton don't have access to. And then others have akinete, which are resting cells that hang out in the bottom of lake sediments through the winter and then rise to the surface and can start growing immediately when conditions are more favorable. Unfortunately, cyanobacteria blooms are increasing in frequency, magnitude, and duration globally. And this is really the result of two factors, eutrophication and climate change. Eutrophication is the excessive enrichment of our ecosystems, in this case, our freshwater bodies, with nutrients. So often we talk about nitrogen and phosphorus, and typically this is through human activity. So talking about excess nutrients a little bit more, where are they coming from? So these excess nutrients come from anywhere in the watershed and come into our water bodies through precipitation and stormwater runoff. So this can be through any use changes. So urbanization can bring in nutrients, agriculture and livestock can, as can just our residential areas on lake fronts. So through runoff from your roof, your driveway, our roadways, unmaintained or failing septic systems, and beautiful green lawns that may be dumping into our lakes. Climate change is also contributing to the increase in cyanobacteria blooms. So with this, our atmosphere is warming, which means our lake's temperature is also warming. The epilimnion, or that surface section of the water of our lakes, is getting warmer in New Hampshire. We're also seeing periods, shorter periods, if we're getting any ice cover in our New Hampshire lakes, which means our lakes are staying stratified for longer based on temperature, and there's a longer growing season. So for some lakes, that means the hypolimnion, the very bottom deep waters, are getting into situations where they have low oxygen or anoxia, which can make the environment uninhabitable for some aquatic life and can also create a situation where we get internal loading, which is when phosphorus that's previously been bound to lake sediments is released into a form that cyanobacteria can then pick up and use for growth, and that can fuel their blooms as well. Also in New England, we're seeing changes as a result of climate change in our precipitation patterns. So instead of having more frequent storm events that bring in regular trickles of nutrients, we are having periods of drought and then huge storm events that bring in large periods or large flows of nutrients all at once. And because of the competitive advantages that I spoke about, it tends to be cyanobacteria that take advantage of this pulse of nutrients and grow out of control into blooms. So once we have a bloom of cyanobacteria, they're extremely dynamic. So these pictures here are all of different cyanobacteria, so hopefully highlighting how dynamic they are in their appearance. They can range in color from deep green, light green, blue-green, yellow, black, white, etc. And they can look like spilled paint, general cloudiness in the water, ribbons of material, or strange chunks of material. It's common to see time of day variation with blooms. They tend to surface during calm periods, like in the morning and the evenings, and then during the day it may look like they have passed, but instead they're just physically mixed back into the water to a point that you might not be able to see it as well. So that's something to keep in mind if you're experiencing a bloom on your water body. It's also common for this material to move around a water body. And something else is that where you see cyanobacteria blooms happening is not necessarily where the problems that are causing those blooms are occurring. This material does grow lake-wide, even in the deepest part of lakes, and then gets moved around to different shorelines based on wind, wave, and boat action. Unfortunately, blooms are also dynamic as far as their length of duration. So sometimes they'll pop up and exist for just a couple of days, and on the other end of the spectrum, they can last for almost entire summer seasons. So unfortunately, this isn't something we're able to predict, so that can be challenging as well. So why are blooms bad? So when all of this biological material eventually decomposes, that process takes oxygen out of the water and can create an environment that is uninhabitable for aquatic life. So you're seeing a picture of a fish kill here. So that is problematic in itself. Then there's the issue that cyanobacteria, many of them, can produce a range of toxins. And when they are in bloom form, the concentrations of those toxins can be elevated enough to pose a negative health effect on humans, wildlife, and our pets. So in addition to blooms being very dynamic, the toxicity as far as the type of toxin and the amount of toxin is also extremely dynamic and can change rapidly over the course of a bloom. So that makes what we're doing here in trying to communicate about risk and cyanobacteria blooms a really challenging thing to be doing. So to talk about cyanotoxins in a little bit more detail, this chart here is showing you a list of cyanotoxins, and it is pretty long already, but this is by no means an exhaustive list. There are many, many different types of cyanotoxins out there. They all vary as far as their mode of action goes and the resulting symptoms that you may have after exposure, but there are some major categories. So to highlight those categories, there are neurotoxins, hepatotoxins or liver toxins, dermatoxins, cytotoxins, and genotoxins. And then there are also tumor promoters. So as you can see, some of these toxins are exerting their mode of action in several different ways. Microcystins we'll talk about in more detail as I will also talk about cylindrospermopsin. These are two more commonly found cyanotoxins. So many of these toxins exert both acute and chronic toxicity in humans, wildlife, and pets. Children and some individuals tend to be more sensitive to exposure. Certainly, if you have a pre-existing health condition, they may affect you more. The symptoms that you may have as a result of exposure vary from mild to very severe, and it depends on the type of toxin you're exposed to, the route of exposure, and how much you've been exposed to. So it can range from dermal, eye, nose, mouth irritations, general malaise, fever, nausea, vomiting, diarrhea, gastroenteritis. The neurotoxins can cause tingling, numbness, and seizures. And in very severe cases, we can see nervous system and organ failure, and in the most severe case, death. So this is much more likely with pets or animals that will consume more of the material to have enough of this exposure to cause those severe situations. So in recognition of two of the more commonly found cyanotoxins that I highlighted before, microcystins and cylindrospermopsin, the EPA has established health recreational guidelines as well as health advisory levels for drinking water and just recreational water. So we'll be referencing those further on. So microcystins is one of those. This is the most common cyanotoxin found worldwide and also in New Hampshire. It is a potent hepatotoxin and tumor promoter and can cause both acute and chronic toxicity. So this diagram here on the left shows the different ways that this toxin can affect the human body. So to highlight those, it can affect the lungs, the liver, the immune system, the stomach, the small intestine, and the colon. And unfortunately, microsystems are extremely stable compounds, so they can exist in the environment independent of cells from 4 to 14 days. And also, unfortunately, 3 of our 4 most common bloom-forming cyanobacteria observed in New Hampshire are capable of producing microcystins. Cylindrospermopsin is the other one that the EPA has recognized. This one is not found as frequently in New Hampshire freshwater. It is toxic to multiple organs, is also neurotoxic and genotoxic. So to highlight the same graph here, it affects the lungs, liver, kidneys, immune system, stomach, small intestine, and colon. So clearly a heavy hitter of a toxin. This one's interesting in that it's not always cell-bound. It's actually released into the water column during active cell growth, which is a bit of a difference from most cyanotoxins. And this one is also stable in the environment and can persist independent of cells. And again, unfortunately, 3 of our 4 most common bloom-forming cyanobacteria are capable of producing cylindrospermopsin. That being said, we don't tend to see it as frequently in our bloom samples. To highlight some of the neurotoxins, there's anatoxin A and guanotoxin, formerly known as anatoxin AS. So anatoxin can cause acute toxicity. It's actually known as very fast death factor. It's a very potent neurotoxin and inhibits a really important neurotransmitter. So if you're exposed, you can see seizures and death. And again, this is much more common to see in our dogs, unfortunately, and other animals to ingest enough of this to have this result. Yeah, so pretty intense toxin as well. So this one highlights, can affect the brain, the central nervous system, but also the peripheral nervous system, which is how you get the seizures from exposure to this toxin. Guanotoxin is kind of similar, also affects a neurotransmitter, and this one can cause excessive salivation, tears, urinary incontinence, muscle weakness, twitching, convulsion, and respiratory distress. Unfortunately, all 4 of our most common bloom-forming taxa in New Hampshire have the ability to produce anatoxin A, and Dolichospermum, which is our most commonly observed bloom former in New Hampshire, is capable of producing guanotoxin as well. So something to pay attention to and think about when we talk about blooms. So how are we exposed to cyanotoxins? So exposure can happen through ingestion. That is the primary route of exposure. So this could be through your drinking water, through recreation. Even as adults, you can't help but ingest a little bit of water as you swim. We know our food sources can also be contaminated. Some of these toxins can accumulate in fish tissue, for example, and some of them are even bioaccumulating as it moves up the food web. There's also evidence that the material can just be on the surface of vegetables or accumulated in the tissue of vegetables. And some folks take cyanobacteria supplements, or if you have a green drink ever or go to a smoothie bar, you may see the option to add spirulina or aphanizomenon, and these are cyanobacteria. The FDA has started regulating this, so this is something to pay attention to if you enjoy the occasional green beverage. They do occasionally issue recalls for products that you can find at normal grocery stores, so definitely something to pay attention to. Inhalation is another potential route of exposure. This can be through recreation or showering with lake water, skin contact through swimming, boating, water skiing, and eye contact similarly, swimming, recreation, or potentially aerosols. Okay, so now that I've talked about cyanobacteria in general and a bit more about cyanotoxins, we're going to shift gears and talk about self-risk assessments, reporting to our program, and how the CyanoHab program here at NHTS works. So through this, we'll be building kind of a flow chart of different actions and the actions that you can take as a member of the public, a lakeside resident, or just someone who recreates on our lakes in New Hampshire. So those actions are taken, are shown in blue that you can take. And the actions that my program will take are shown in green. So as we build this, that will make a little bit more sense. So ideally, this all starts with a self-risk assessment. So every time you go to a water body and before you recreate or let your pets in the water, we're hoping you're doing this self-risk assessment. So really, this is looking at the water and looking for any unusual growths, discoloration, flecks, plumes of material, ribbons on the surface, etc. So it's looking at the water, but also part of a self-risk assessment is staying informed and keeping up with the information that we're putting out there to communicate about cyanobacteria. So focusing in on the looking at the water, it's important to know what cyanobacteria blooms look like and to consider what other lookalikes may look like. So we're going to focus on this part of a self-risk assessment first on the next handful of slides. So these pictures are all of planktonic cyanobacteria blooms that we had submitted to us last year from across the state. So planktonic just means the cyanobacteria is growing lake-wide throughout the entire water column and is getting moved to different shorelines based on, again, wind, wave, boat action, lake morphology. So just good to look at these, build your understanding of the diversity of how they may present in your water body and know what to look for. Just to highlight a couple of unique cyanobacteria that we see in New Hampshire, there is Gliotrichia, which looks like little round balls in the water. And we do see this in some of our deeper, cleaner lakes. So if it looks like the density shown in this picture here, we like to say that looks like a starry night, in which case recreation would be low risk at that point. But if it starts looking like a galaxy, like in this picture and some of these other pictures, and certainly if it's forming a surface skim like in this bottom right picture, then you would want to not recreate at that point and wait for the densities to change. Gliotrichia tends to stay more well distributed through the water column and doesn't frequently form these blooms, more bloom-like material, but if they do, you just want to avoid it and certainly report it to us. Benthic cyanobacteria is also different from our more typical planktonic bloom-forming cyanobacteria. This can also appear in a range of different ways. So there can be fluffy balls of material, sludge-like material that comes up along the shoreline, or different crusts and mats of material. So benthic cyanobacteria is a bit different. You'll see if you have this on your water body, we do communicate about this a little differently, and we hope to continue developing and formalizing how we're dealing with benthic cyanobacteria as this program matures and progresses, and as the scientific community figures out how to communicate about benthic cyanobacteria as well. So switching gears and looking at some of the common lookalikes that we get reported with folks thinking that it's cyanobacteria, but they're not actually cyanobacteria. So probably the most frequent one that we see is green filamentous algae. So this is harmless in that it doesn't produce any toxins, but it can grow to nuisance levels, as you can see on these bottom pictures. Some diagnostic features to tell it apart from cyanobacteria are the bubbles that get trapped in the material. That's just from photosynthesis and the gases that are released. But if it was cyanobacteria, there isn't that extra structure to capture those bubbles. So another thing you can do is the stick test. So you see these two pictures in the middle here. Literally just put a stick in the material, lift it up. If there's any goopy, slimy material hanging off the stick, then it's very likely green filamentous algae, and that's harmless. If it were to coat the stick and not have any of this extra structure to it, it may be cyanobacteria, so you'd want to report that to us and stay out of the water. Duckweed is another commonly mistaken organism, not cyanobacteria. It can grow to nuisance levels. As you can see, it looks like an aggressive bloom on these water bodies, but if you look closer, maybe put a stick through it again, you can see that there are tiny leaf structures and tiny root structures. So cyanobacteria are microscopic organisms, so they don't have any of this extra structure to them. So that's how you can tell those apart. Iron bacteria is another one. This can present as this orange material in these top two photos, or more of what looks like oil slick, which is a biofilm produced by these organisms. This is not cyanobacteria, doesn't produce toxins, so that's good to know. If you are trying to distinguish it from an oil spill, again, grab that stick and tap it. And if it fragments into these little pieces, then it's just a normal biological phenomenon and not an oil spill. Pine pollen is another one that is kind of tricky. So if you are seeing pine pollen everywhere, if it's on your cars, coming in your windows, you may see it in your lake shores like these pictures. I say it's tricky because pine pollen is a shot of nutrients into the ecosystem, and sometimes we do see cyanobacteria mixed in with this material. So best practice with any of these scum photos that I'm showing you that are not cyanobacteria is still to avoid that area, keep your pets out, keep the kids out of the water until it passes, and it should pass eventually. If it starts to look like this picture that just appeared here with some green around the edges, that is cyanobacteria mixed in. At that point, you would definitely want to report it to us, and we'll try to evaluate a sample. But overall, with all of this, if there's any doubt in your mind that it could be cyanobacteria, the best choice that you can make is to stay out of the water and report it to us. Lake foam is another natural phenomenon. If you see this, it doesn't mean your neighbor has dumped detergents into the water. This is just from the degradation of biological material. Some cells can release surfactants into the water, and when air gets mixed in, it just creates this bubbly, frothy material. So this will also pass on its own. It inherently is not posing a health risk to you if you were to interact with it. But again, best practice, any surface scum that you see, when in doubt, stay out. And just finally, there is at the top of the presentation, I mentioned other types of phytoplankton. So this is a chrysophyte bloom, which is a golden algae. And it can occasionally bloom in New Hampshire. We tend to see it more in the spring. It looks different from cyanobacteria in that it doesn't form a surface scum that we see with cyanobacteria, but can form these big clouds that stay under the surface of the water. So if you see this, definitely report it to us. It is really hard to distinguish. We have to look at it microscopically to identify what it is. One feature that you may notice about this, if you see this blooming on your water body, is that it smells bad. It produces smell and taste compounds, so that might help clue you in. Some cyanobacteria can also smell bad, so not a totally distinguishing feature, but something to be aware of. Okay, so we just talked about the first part of this self-risk assessment, which is all about looking at the water, knowing what cyanobacteria looks like, and considering common lookalikes. The second part of our self-risk assessment that we're hoping everyone will do is staying informed. So talking about that on the next few slides here. So this is all about staying in tune with the resources that we have out there. So we have a really great resource, the Healthy Swimming Mapper. We have a way that you can sign up for emails for a specific water body or sign up for our statewide status emails that summarize the conditions across the whole state. So I'm going to spend the next few slides talking about this. So this Healthy Swimming Mapper, we just redesigned it last year thanks to my incredible coworker, Michelle Condon, who did all the legwork for this. It's great. It's a really great resource. It has so many different features to it. It's also the first resource that we update, so it's always checking for updates from us, and it will be the most up-to-date resource that you can find from us. The advisory or warning areas that we have are red symbols on the map. Alerts are yellow, and I'll talk about the distinction between those two in a few slides here. But if you're interested in those, you can click on the symbols there or click from the dashboard summary on the right-hand side of this map, and the map will zoom you to that area to look at that lake in more detail. If you don't see a symbol, you can always use the search button, and this also has a bunch of other resources linked from this page as well. So if you click on a lake that has a warning, it will bring you to it and bring up this dialog box that has a bunch of information in it. So it lists the current status, the town of the water body, the most recent sampling date the status was set, a bloom description, the initial cyanobacteria identified, and the initial total density. We don't update the map with when we resample, but if it's still listed as an advisory on the water body or warning on the water body, then it's still an active warning. If there had already been a warning through the current season, the dates would be listed in that column there. And also there will be a link for every water body to access the historical cyanobacteria events, so both warnings and alerts, and we are working on updating those with last year's data currently, and that's coming soon. There's also pictures of the current bloom material so that you can know exactly what the material you should be avoiding looks like in that moment. We also added this year this link that you can use to sign up for cyanobacteria emails for that specific water body. So a really easy way to stay in touch with our more detailed sampling updates. So if you click that link, it will bring you to this form where you can sign up for as many water bodies as you want. Just list them all there. The instructions on the top tell you how to do that. And this is really us trying to target specific communities to share information, a little bit more detailed information with folks that really need it to prevent exposure. We also use these email groups to coordinate resampling and provide more details on what we're seeing and how the event is progressing. Once you finish submitting this form, it will bring you to an option to sign up for this weekly update. We also have unique links for that if you're only interested in the broad overview, but this is shared really widely through our constant contact platform. It's posted on our website, and it's emailed if you sign up for the list, and it's emailed to the press. So this just broadly summarizes where the active warnings are, active alerts, and where warnings have been removed within that week. And we issue these every Thursday through the growing season. Okay, so we've talked about the self-risk assessment and its two primary components, looking at the water, just a common sense approach to evaluating the situation for cyanobacteria, and also checking our resources. So we're really lucky in that cyanobacteria are very visual, so definitely take heart in your self-risk assessments and know that you can do a really good job protecting yourself and your family and your pets from exposure without even getting a cell count or a sample from us or to us. So definitely get used to doing this self-risk assessment and know that it's a really valuable tool for protecting yourself and your families. So say you've done this and it does look like it's potential cyanobacteria. So the first line is, when in doubt, stay out of the water and keep the pets out. But we also hope that you report it to us. So how do you report it to us? So if you're following along, you can use your phone camera to scan this QR code to get this bloom report form on your phone right now. But just to show you some screenshots, it will start out with some picture examples of what cyanobacteria blooms may look like. And then the rest of the form is all standardizing the information that we get from folks when they need to report blooms for us. So we ask for information about the bloom, pictures of the bloom, which are super helpful in figuring out the situation and how, how far along the bloom is, et cetera. And we'll ask for water body information if you're able to help with sampling, and just a little bit of information from you to make sure we get the information we need and can share that to you. So once you've submitted, you'll get a confirmation page. It'll remind you, when in doubt, stay out of the water, and it lets you know when we are in operation, so Monday through Friday, 8 through 4. If you submit outside of those hours, take confidence in your self-risk assessment, stay out of the water, and know that we'll get to you as soon as we can. There's also a bunch of helpful links at the bottom of this page that you can use to get a little bit more information before you may hear from us. So you've reported it, and then this is where I come in and I will review the report and determine if we need to collect a sample. So if you're able to collect a sample, that is a huge help to us. If your water body is blooming, there's a bunch of water bodies across the state that are also blooming. So we're running around trying to get everywhere. So if you can collect a sample, that's a huge help. If not, we will come and sample the water for you. If you can deliver it to DES, that's even better. If not, we'll come pick it up. But either way, this all ends with a sample coming to DES, and it's time for me to do an analysis on it. So again, if you're able to collect and deliver to us, that will not only help us as we try to get around the state responding to bloom reports, but it also helps you in that you will get results faster if you're able to deliver a sample to us. So that's just something to keep in mind if you have the ability to do that. Otherwise, we will come out and get a sample eventually when we can. So what's cool about this bloom report form is it's adaptive. So we do have a question, are you able to collect a sample? And if you respond yes, then you'll get the sampling instructions. So if you're going through this, you can take a screenshot at that point and have that as a reference point for later. But otherwise, it's really simple to collect a sample. Wear gloves, use the proper PPE, any plastic container will do to collect the sample. And then you just want to skim the surface of the water in the area of the most concentrated bloom material so that we can characterize risk with the most or the worst presentation of the bloom. Any hard plastic or glass container will do, and samples should be stored on ice or in a refrigerator until they can be delivered to us. You can also fashion a sampling pole. This is what my interns use when they go out sampling. And this just provides a little bit of distance between you and the bloom material. So it can be as simple as a bottle taped to the end of a stick. So just another idea if you're able to do that or interested in doing that. As far as delivering a sample goes, the form will also give you instructions on that, but it's simple. It's just coming to the Jody Connor Limnology Center at our DES office in Concord. So this all ends with me getting a sample to analyze. So to bring it back to the EPA guidelines for microcystins and cylinders per mopsin, through studies, that level of 8 micrograms per liter has been related to a density of 70,000 cells per mil of cyanobacteria. So in New Hampshire, this is what we use for our recreational threshold. We do take subsamples from some bloom samples for future toxin analysis. We use the technique called an ELISA, but we don't run the program based on toxin analysis because it is expensive to run these quantitative tests. It's very time intensive, and the results would really be too delayed to inform public health in a meaningful way. It's also worth remembering that chart that I showed you with a bunch of different cyanotoxins. There's many cyanotoxins out there. So if we focus the program just looking at one of them, we may be missing the risk associated with some of those other toxins. So instead what we do is a microscopic analysis. I'll identify what cyanobacteria are present and enumerate their presence. If it is above that 70,000 cells per mil threshold, below, I may issue an alert. If it is above that threshold, I will definitely be issuing a warning. So a little bit more about an alert versus a warning. So an alert is just a statement to be on the lookout for cyanobacteria. We issue these under a couple of different scenarios. So it could be that the cell density is just below the warning threshold, but we think it might develop into something more significant. We'll issue these if the bloom has passed by the time we're looking at a sample. So for example, if you collect a sample for us on a Friday afternoon, and we aren't able to look at it until Monday morning, then, but by then the bloom has passed, then we may issue an alert to just say, be on the lookout. This could reappear in the next couple of days, or it may have passed, but we just want to get the word out there. Sometimes we also issue alerts based on a photo or a description of the bloom prior to sampling when it really looks like it could be cyano, and we just want to get the word out there quickly to communities. So if we issue an alert, we only will resample if residents inform us about the continued presence or changing condition, so worsening condition typically, and alerts are active on our healthy swimming mapper for a week. A warning is a much more formal statement that the lake water is currently unsuitable for wading or swimming, and to not come in contact with the bloom material, especially keeping children and pets out of the water. So again, this is always issued if the density exceeds 70,000 cells per mil. When we issue warnings, we do resample lakes weekly until the bloom has passed. We really like to see the cell density decline very low, below the recreational threshold, to make sure that the bloom doesn't just reappear the next week. It ends up being more confusing for communities if we have to issue and remove and issue warnings than just keeping them for a little bit longer to make sure it's fully passed. Another important point to make is that we need samples to be collected under representative conditions. So if we're resampling during a rain event, or if it's really windy that day, the cyanobacteria material that typically forms surface scums may be physically mixed back into the water, so we're not able to see if they're still present at unsafe levels. So if this is the case, we appreciate your patience with this and know that we'll be back out the following week to confirm if the cyanobacteria has really passed. and if it has, we'll remove the warning at that point. So again, we do issue these as lake-wide warnings. We unfortunately are not able to characterize risk in real time around a water body when there are cyanobacteria bloom events. As I've mentioned, they're very dynamic events, so we just issue them as that folks anywhere around the water body are made aware of that and know the potential risk of recreating. So we also don't close water bodies when we issue warnings, but we do advise against recreation. So if you choose to recreate during a warning, know that there is definitely some risk associated with that, and know that the risk is a lot higher if there's any visual presence of cyanobacteria. So even just, but certainly any blooms. And know also that children and pets are most vulnerable, so keep that in mind if you're choosing to recreate at any point, but certainly if there is a warning in place. Okay, so now that we've talked about how to do a self-risk assessment and you have some understanding, hopefully, of how our program works and what we're doing with samples here at DES and how we're communicating about it, I'm going to use the rest of our time today to talk about cyanobacteria trends over time in New Hampshire as collected through this program. So this map here is showing you all of the different unique water bodies where we've issued cyanobacteria warnings since the program was started. As you can see, they've been issued across the whole state, north to south, east to west. And unfortunately, every year we do recruit new lakes to this list. To date, we've issued at 122 different lakes. This graph here is showing you years of program operation on the x-axis and total number of warnings on as the bars, warnings or alerts. So the warnings are red bars, the alerts are yellow. The fewest number of warnings we've ever issued was the first year in 2003, just one, and the most we keep breaking every year, but we broke it yet again last year with 69 warnings in 1 year. Over the last 5 years or so, on average, we've issued around 41 advisories. We started issuing alerts in 2020, partially in response to the pandemic, but we also just needed a faster way to communicate about cyanobacteria. So those have been very helpful for that. So the fewest we've ever issued was that first year, 34, and the most was 2021, 87. We are ironing out our alert history, so these numbers may change a little bit as we figure that out. So some water bodies do have, sorry, I gotta move my panel here so I can see my slides. Some water bodies do have multiple warnings and alerts within a single year. So for example, in last year, there were 69 warnings total at just 47 water bodies. Unfortunately, we have seen a statistically significant increase in warnings since 2003. And as I talked about at the beginning of this presentation, this is a result of eutrophication, so excess nutrients, and climate change. But something else to keep in mind is the data collected through this program is it is a reaction-based program, which means samples are primarily collected when they're reported to us. And we have definitely seen increased public awareness about cyanobacteria in general. So we have been observing more reports, and more reports leads to more warnings, which kind of seems like a bummer, but overall warnings are super important. They keep our people and our pets safe, so they're important to keep reporting to us. Okay, so this graph here is showing you on the x-axis, we have the different months that we've issued advisories or warnings, and the bar is the total warnings ever issued in program history within those months. So as you can see, we've issued warnings from May through October. The most are issued during peak summer months, so August and July. Colder temperatures do mean less recreation, and so with fewer eyes out there, we get fewer reports. We do know cyanobacteria are so good at what they do, they can bloom under ice. So if you ever see this, we want to know about it, report it to us so that we can document that it's happening. Another question I always get is, how long is this going to last when I issue warnings? So this graph here is showing you, again, year of program operation on the x-axis, and then the average number of warning days within that year. So over the last handful of years, roughly 2018 to 2023, the program's been run a bit more consistently. So on average, warnings last for around 24 days. The shortest in that time frame was 2 days, and the longest was 132 days. And the duration of warnings and bloom events varies greatly and depends on a bunch of different factors. It depends on the lake morphology, the current nutrient inputs, the legacy of nutrient inputs, and the weather. So many different factors. Unfortunately, we can't predict this. We just have to kind of ride it out together. To talk about last year in review a little bit more in detail, it was a record-breaking year, so there's a lot going on in this graph. So to break it down, the x-axis is again the different months of program operation, and the bar is the number of warnings that we issued within different years. So the different years, 2019 to last year, 2023, are different shades of green. And then you'll see some bars have asterisks on them, and that is the previous program record until last year. So this is highlighting the number of warnings that we had in each month last year. So you can see it was a really big cyano year. So we set the record for May, June, July, August, and October for the most warnings ever issued. We also set the record for the most warnings issued within 1 month. In both June and August, we hit 18 warnings. Prior to that, the most we had ever issued in 1 month was 15. We also issued our earliest warning on May 16th. Previously, it was May 20th, just the year before. And then our final advisory day, the day we lifted our last advisory, was the 14th of December, previously 7th of December in 2021, though that bloom did reappear shortly after. So clearly we're seeing the cyano season is extending in both directions, early in the spring and late into the winter in New Hampshire. So I'm sure we'll continue to see those trends as well. To end out my presentation here, just one more slide to bring it back to the cyanotoxins, which is why we ultimately care about cyanobacteria blooms. So there's a lot going on in this graph as well. I'm going to break it down for you by focusing on 2022. Unfortunately, we don't have the data for 2023. We are actively working on that, so stay tuned for that data as well. So the total bars on this graph are the total number of warnings that we tested for specifically microcystins, so the most common cyanotoxin found in New Hampshire. The green part of the bar is the number of warnings that did not have microcystins present. They were at least below detectable as our test could measure for them. The yellow had microcystins present to some degree, and the red had microcystins present above the EPA recreational guideline. The percentage on the top of the bars is the percentage of warnings that had some level of microcystins present. So the percent has varied year to year from 56 to 73%. The number of warnings with microcystins above the EPA recreational level has ranged from just 1 to 14. 2022 was a middle of the ground year with 6 above. Something to keep in mind though is these data are showing you just the initial bloom toxicity, and we do know that bloom toxicity can change over the duration of an event. So even though it may look like the bloom may not have been toxic based on the first sample, it could have become toxic the week after. So that's something to pay attention to. And in addition to that, if you remember that chart I showed you, microcystins are not the only cyanotoxin, which is why hopefully that sets home we don't run our program based on toxin analysis, but do a cell count instead in hopes of capturing the risk associated with a range of cyanotoxins. We are working on creating new resources. So we have new frequently asked questions that will be updated very shortly. I'm working on recording videos, so similar to the presentation you've seen today, in different segments so that you can choose what you may want to share with a lake association meeting, for example. We're also working on updating fact sheets to have one about sampling and program specifics. We're also working on creating permanent signage that we hope to have at all public access points around the state. So everywhere you've seen the clean, drain, dry signage for the prevention of spreading aquatic invasive plants, we hope to pair with a permanent sign reminding folks to do those very important self-risk assessments and have links so that you can access our healthy swimming map and our bloom report form while lakeside so that we can get the word out about doing self-risk assessments. Oops, sorry. We're also hoping to build our dashboard page. So during the summer season, I often get how many advisories have we had so far this year? So we're hoping to build a page that summarizes that for you so that you can have that information more readily accessible without having to check in with me. And with that, I'm happy to take questions. Don't forget to do your self-risk assessments this summer. If you see something suspicious, stay out of the water and report it to us, and we will be in touch with you about what to do with that. So at this point, Michelle Kahn is going to join me and I'll try to address some of your questions.
Yes, thanks so much. Great job, Kate, as always. So yeah, continue to put your questions in the chat box. We've got great questions so far. We're going to try to get through as many as we can until the 1 o'clock hour. But if we don't get to your questions, we do have your email, so we'll be able to reach you through that as well. So the first question, Kate, that I've received a few questions about the self-risk assessment. The first being, let me get to it here, about kayaking during a bloom and if that's safe to do.
Sure, yeah. So we typically say that you should suspend any recreation during a warning, just as kind of a broad measure to keep people safe. So any sort of recreation that you may come in contact with the bloom material. So with kayaking, at least when I do it, I tend to get the water dripping down my arms. So you can have dermal reactions to cyanotoxins. So that's something you'd want to pay attention to and avoid that type of exposure. But great question. I mean, typically boating should be less exposure. We do know that in some cases, cyanobacteria and their toxins can aerosolize, so that may be another component that you'd want to consider if choosing to recreate during a warning.
Another question regarding the self-risk assessments. Question about Lake Mascoma, which is a very long lake. Is there danger if a bloom is occurring in one side of the lake, but you're recreating in the other part?
Yeah. So there's a lot of nuance to these questions here and what people may find more comfortable. So again, our blanket statement is avoid recreation in the whole water body because we can't characterize risk perfectly. If you choose to recreate, the risk is probably not going to be zero. This material does grow lake-wide, even in the deep section of the water, so it could be present at lower levels that you may not be picking up visually. But that being said, recreation is a lot safer if you're choosing to do it in areas where you don't see any evidence of cyanobacteria blooms. And also know that sometimes it can just look like tiny little flecks that if you're not used to looking for it, you may not catch it. So just pay really close attention to the water and know that conditions can change really rapidly. So certainly if you have children or pets near the water, pay attention to that.
Great. Switching topics a little bit, broad question, is there any way to control the cyanobacteria blooms?
Yeah. So as I mentioned, a lot of cyanobacteria blooms are happening because of eutrophication, so excess nutrients coming into the water body. So anything that you can do if you live on the lake edge to reduce stormwater coming into the water body is a great move. Plant shoreland vegetation to take up nutrients before they enter the water body is helpful. Get your septic systems maintained regularly and inspected regularly. There's a lot of different things. So New Hampshire Lakes also has a program where you can get your property evaluated for how lake friendly it is, and they will suggest specific opportunities for you to improve if needed to prevent nutrient runoffs into our water bodies. So that's one way to control cyanobacteria. And with a community coming together around a water body, it can have an impact on preventing future cyanobacteria blooms and moving forward.
Similarly on shoreline living, so someone is interested in the fallen leaves on their shoreline. Besides the aesthetic effects, is it best interest to rake those out of the lake to prevent cyanobacteria blooms later in the season, or what do you recommend about fallen leaves?
Yeah, fallen leaves are a more natural phenomenon, so that's certainly not going to be causing cyanobacteria blooms. It's more of the anthropogenic, so human-caused nutrient inputs that we should be trying to control. The ecosystems are balanced. We don't typically see cyanobacteria unless there's some larger imbalance in the ecosystem. So wouldn't worry about your lake as far as leaves go.
Next question, should people report if they or their pets get sick after recreating in a lake?
Yeah, that's a great question. So we do track that. We are working on developing a new health illness report form that should be live this year. But if you follow through and submit through our Bloom report form, a link to submit a health incidents will also come up. And we do have links as well on the Bloom report form for the CDC guidance that you can bring to a healthcare provider or a veterinarian in case that's helpful, and to inform what might be done in that case as well.
Great. Next question. Are any environmental or other factors found in the reasons for 2019 and 2023 being higher? I'm assuming that's having to do with advisories and alerts.
Yeah, so last year we had a lot of big storm events, which, as I mentioned at the top of the presentation today, can bring in these huge pulses of nutrients that we wouldn't typically see, and all at once, which the cyanobacteria are so good at taking advantage of. That being said, some water bodies bloom more in years where there are droughts, and some water bodies bloom more in years where there's a lot of rain. So each lake is very unique in their response, and subsequent issues with cyanobacteria are also very unique. So it's hard to predict statewide. So there were a couple of environmental factors. I mean, also the fact that we had not a long period of ice cover before last year certainly played into it as well, we believe. So there's a lot of confounding factors, hard to point to just one. These are dynamic events, so hard to predict how it might be going forward. But again, I want to point out that this is a response-based program, and cyanobacteria have definitely been in the news more, and certainly in kind of the attention of New Hampshire. So we are getting more reports because of that, which is great. We want people to be aware of cyanobacteria and know to do their self-risk assessments, but that could be influencing what looks like an increasing amount of cyanobacteria warnings in New Hampshire.
So prior to the bloom, are there any precursors of like measurable factors like temperature, pH, that you are aware of?
That's a great question. Unfortunately not. Blooms are really hard to predict. And certainly you're not really able to unless you have some water quality measuring opportunities. But they pop up really fast. So even when folks are trying to predict blooms, it's a really hard thing to do. So, there isn't a quick way to do it. We are, again, lucky that they're very visual. So typically, if you're in tune with what's happening in the water body, you can see flecks first starting to develop. And then in a couple of days, it may turn into a more dramatic bloom like you're seeing on the slide now.
Great. More specifically about cyanobacteria, what is the typical lifespan for cyanobacteria, and can their toxicity be retained after they're present?
Yeah, great question. So the individual cells themselves don't necessarily live for that long, but the bloom is just continual cycles of cells building up and into that biomass. The toxins, as I mentioned, can exist independent of cells. So particularly the two that I highlighted, microcystins and cylindrospermopsin, they can exist in the water independent of cells for up to 2 weeks until they're degraded by different normal environmental factors like access to sunlight, et cetera, will eventually break down those toxins. But yeah, they can persist independent of the cells. So if you're particularly conservative, you may wait till after the bloom has passed visually before choosing to recreate again.
Great, thank you, Kate. Can you differentiate between an advisory and a warning?
Sure, great question. So advisory and warnings are the same thing. In response to the Cyanobacteria committee we had in creating the New Hampshire Cyanobacteria plan, we got the feedback that advisory was probably not a strong enough word to get people's attention. So we have switched gears and are calling them warnings. So we do try to pair those together in written communications to try to make that clear, but I'm glad I was able to address that today. So advisories and warnings are the same thing, and then alerts are the step down from that situation.
Great, a more specific drinking water question. So these folks use lake water for their cottage. Is there a type of water filter they can use to remove cyanobacteria?
Yeah, so home filtration systems are really tricky and likely won't be able to do a good job at filtering out cyanobacteria and their toxins. So some filters may be able to take out the cells themselves, but the toxins themselves, if it's dissolved in the water, are not really able to be taken out by a filter easily, and certainly not the filters that you're probably using in your home systems. So the EPA has a lot of good guidance on water treatment for cyanobacteria. So you can feel free to email me and I can send you those links. But our general statement is that you should not be using the water in your house if there is an active warning or if you can see any evidence of cyanobacteria. And know that boiling water will not reduce the toxins. It actually can increase the toxicity of the water. So something to pay attention to, and know that, yeah, just don't use the water in the house if there's warning. And, if you're interested in pursuing this more, there are some resources out there by the EPA that we can share with you.
Great, thank you so much. Question about sample drop off. Is an appointment necessary, and who do you contact to make the appointment, and what about weekend availability?
Great question. So let's see. So the Bloom report form has the information on when you can drop off a sample. So that's all detailed there. You don't need an appointment, but it's nice when you submit the form there's a comment section you can just include, I'm going to drop this off right away, Kate. It will come directly to me. To just the Jody Connor Limnology Center has a receiving window that you can come to. So yeah, just leave me a note if you know that you're going to drop it off immediately. That's always helpful. Unfortunately, we are not open on the weekends. So, take confidence in that self-risk assessment. Typically, you do not need a sample reviewed to know that the cyanobacteria is above the advisory level. If it's present and blooming, like in any of the pictures here, or even to a lesser degree, then it's very likely above the warning level, and you should be staying out of the water. So we will eventually look at a sample, and we appreciate that coordination, but unfortunately, we're not open on the weekends to look at samples. But know the power of the self-risk assessment and that you don't necessarily need us to make the right decision to protect your family and yourself and your pets from cyanotoxin exposure.
Great. Kate, do you anticipate more blooms this year due to the early ice out we have? For example, Lake Winnipesaukee already reported ice out this.
Yeah, I'm bracing for another busy year. Yeah, we did see that last year as well, and it did seem to have an impact on our bloom situation. It was a very busy June last year, as you may remember from the graphs. Um, but we'll see. We will see how the year progresses. As I mentioned, with less ice cover, there's just a longer period of time for cyanobacteria to accumulate to these dense levels. So we could have another busy record-breaking year ahead of us.
Great, thank you so much, Kate. There was lots of questions asking if the presentation will be available later. That will be available on the DES website. And we are closely approaching the 1 o'clock hour. Kate, do you have time for more questions, or do you want to end it at that?
I could take a couple more if that's easy enough.
All right,
Maybe a few
I'll
more
get
and
a
then
few
we'll wrap
more.
it up.
How old can a sample be in order to be submitted?
That's a good question. So we ideally want to get them fairly quickly. But if you collect a sample on a Friday afternoon, it's no problem for us to then look at the sample on Monday, so long as you're keeping it refrigerated or on ice. That will prevent degradation of the sample.
Great, thank you. Let's see. What about using lake water to water lawns or flowers, gardens?
Yeah, great question. So using it to irrigate your lawns and flowers is totally fine. If you're choosing to irrigate oh gosh, I'm sorry, folks, my lights just went off in my room. I guess it is time to end. Let me wrap up this question here. So if you're choosing to irrigate your vegetable gardens with the water, there is some evidence that cyanotoxins can be accumulated into the vegetable tissue itself, and it can also just be on the surface of vegetables. So make sure you're washing the vegetables well if you do choose to do this, but finding an alternative source to irrigate your vegetables with is a better option.
Great. So I guess with the lights turning off on Kate, that should be the sign of the end of the webinar. Again, if we didn't get to your questions, we'll try to get back to you via email in the next few days. Thank you all for joining this webinar, and have
Thank
a
you,
great rest of
everyone. Take care.